RNA. So transcription is the process of making RNA from the DNA template. You'll hear the word template thrown around. So if you remember, we have our chromosomes, and when the chromosomes are all condensed, the DNA wraps around histones. But usually transcription occurs in the cell without it having to be condensed into a chromosome. And so if we took a strand of DNA, sorry, my DNA isn't perfect. We know that on our chromosomes we have genes. And so a certain location on our DNA molecule is going to be the location of a gene. And a gene is something that codes for our traits. And molecular speaking, genes code for proteins. They do other things, but proteins are the main thing we're going to talk about. So only this location of the gene that's going to be transcribed is going to be involved in the process. And so the first thing that happens is the DNA molecule needs to unwind in the area of the gene. The DNA molecule unwinds where the gene is. There is a part of the DNA at the beginning of the gene that's called the promoter. And that's just a starting place on the gene. The next thing that has to happen is that the DNA molecule needs to unzip at the location of the gene. So the DNA molecule unzips at the location of the gene. So just this area here of our DNA strand that's part of a chromosome. And so over here at the bottom, I have unzipped my gene with some DNA nucleotides. And here's where the 3' and 5' carbon are going to come into play again. Only the 3' strand is red. So only the 3' strand is red to make RNA. The other side just hangs out and waits for everything to get finished. So remember that we have this promoter area on the gene, and that tells RNA polymerase, not DNA, because this time we're making RNA. It tells RNA polymerase where to start. And RNA polymerase is going to bring RNA nucleotides. And if you remember, they are going to be different than DNA nucleotides. RNA has ribose instead of deoxyribose, and it has the nucleotides adenine, guanine, cytosine, not thymine. Instead of thymine, it has uracil. So uracil replaces the thymine. So RNA nucleotides have ribose instead of deoxyribose, and we have U's instead of T's. So RNA polymerase is going to start building the polymer RNA. And so it's first going to bring in an A, and then it's not going to bring in a T because there's not any T's in RNA. It's going to bring in a U, then it'll bring in a G and a G and a G. It's not going to bring in a T. It's going to bring in a U, a G, a C, an A, and it's going to keep on building the RNA nucleotides. So it's going to keep on building the RNA. When it gets to the end of the gene, the RNA is going to break off away from the DNA template. So you have your 3' strand of DNA. You have your RNA that's being built, and then when it gets to the end, the RNA falls away. Remember, you have your 5' strand over here just hanging out, not doing anything. And so the RNA falls away from the DNA molecule. And then the DNA molecule, when it gets to the end, is going to re-zip up. So it's going to re-bond those bases together, and then it's going to coil back up. So it's going to re-zip and then rewind. So now we have our original DNA molecule the same as it was, and we have a single-stranded strand of RNA nucleotides. So now we need to look at the difference between RNA and DNA. DNA is double-stranded. RNA is single-stranded. DNA has deoxyribose. RNA has the sugar ribose. DNA has adenine, thymine, guanine, and cytosine. RNA has adenine, uracil, guanine, and cytosine. So transcription is a process in which we make mRNA from the DNA template, and only the 3' strand is read.